Okay, so now we are going to get into what we call the tubular component of the nephron, because these are the P tubes, the tubes that have things that are going to eventually end up getting urinated out. So let's go through the big picture first, and then we'll look at it in more detail. So where the tubular component of the nephron starts is this thing that is snuggling around the outside of the glomerulus. Now, the glomerulus is actually a blood tube, but there is a structure that snuggles the outside of it called the glomerular capsule, or the renal capsule, or the Bowman's capsule. And what that is that is the first place that you have a microscopic P tube. Okay, and we'll come back to that and look at it in more detail in just a second. But then that is actually going to lead to the first tube that is close to the Bowman's capsule, or the glomerular capsule, and that is called the proximal because it's closest, convoluted because it's curvy, tubule as in little tube. So the proximal convoluted tubule. Then if you follow the proximal convoluted tubule, what it's going to do is it is going to lead to the nephron loop. And the nephron loop will lead back to the distal convoluted tubule. And it's hard to tell proximal from distal unless you follow it here. The distal convoluted tubule will actually end up dumping into this guy, which is called the collecting duct, and we'll come back to that in just a second. So let's go over each part in more detail. Okay, so first thing, glomerular capsule or Bowman's capsule. These are located up here in the cortex, and really what they are is they are snuggling around the outside of the glomerular capillaries. So I want you to imagine that the glomerulus, the capillary thing, is like my hand, and that the Bowman's capsule or the glomerular capsule is basically like I take this hand and I push it into a softly blown up balloon, and I end up with balloon coating each finger, and then a space, and then the outside of the balloon. So that's what you're seeing right here. That's why this structure right here doesn't actually look like simple squamous epithelium, because it is, but it's got a glove on, and the glove is actually the inner or visceral layer of the glomerular capsule, and then I have a space, and then I have the parietal layer of the glomerular capsule. So if you blow this up and look at it more closely, here is what the capillary looks like, and then on the outside of the capillary is the first layer that you run into of Bowman's capsule, or the glomerular capsule. So the capsule is actually a P tube, and the glomerulus is actually a blood tube. So it's basically like a balloon that's like pushed in on one side. You've got the visceral layer that's coating my finger capillaries, and then a space, and then the parietal layer. Now this structure that has both the capillaries and the capsule, this is called the renal corpuscle, and this is what it looks like inside the kidney. The renal corpuscle is really, really important because this is the primary site of filtration. And the reason it's so good at filtration is because that afferent arteriole is much bigger than the efferent arteriole. And because it's really, really porous, the capillary itself is holy. And then between the cells that form the the visceral layer of the glomerular capsule, there are little filtration slits. These cells are called podocyte cells, podo as in feet, and they have little slits between them. So the podocyte cells with the filtration slits. So collectively, this place, both that pushes the stuff out and that catches it, is called the renal corpuscle. Primary site of filtration in the kidney, fluid and small solutes get pushed from the blood into this space between the visceral and the parietal layer. They get pushed through the capillary and then pushed through the filtration slits, and then they're going to end up here. Okay, from there, where they are going to go is through the proximal convoluted tubule. And the proximal convoluted tubule is what attaches directly to the capsule. So let's look. This one actually has the blood tubes taken off so that you can see. So afferent arteriole went in, efferent arteriole comes out. Here is the capsule, Bowman's or the glomerular capsule, and it leads to the proximal convoluted tubule. Why is it called proximal? Because it's close. Convoluted means curvy, and it is a tubule. Now, FYI, way back at the beginning of the semester, you learned that kidney tubules were made of simple cuboidal epithelium. This is what they were talking about when they said a kidney tubule. They're made of this. Does this look kind of familiar? These little guys right here. So the proximal convoluted tubule, often abbreviated PCT, starts up in the cortex, and then it might dip a little bit into the medulla, but it's mostly going to stay up in the cortex. And then what's going to happen is it's going to dip. It's going to go down and then back up. As it goes down and then back up, it's going to become what we call the nephron loop, or they used to call it the loop of Henle. The nephron loop, depending on how long it is, it either dips really far into the medulla or it stays mostly close to the cortex. It is primarily for water reabsorption. So it goes down and then back up. So descending limb first and then ascending limb rises back up into the cortex, and then it's going to lead to the distal convoluted tubule. And the distal convoluted tubule is again curvy and again small, but further away from the capsule. So the distal convoluted tubule will actually dump into what we call the collecting duct. And the collecting duct, by the time you get into the collecting duct serves more than one nephron. So it's actually not considered part of the nephron. It would serve like all of these nephrons would have dumped into it. And the collecting duct is not a part of every nephron. It's basically going to be like collecting from multiple nephrons. And the reason the collecting ducts, or the reason that, back to here, the reason that these pyramids look striped is because the collecting ducts are all running this direction. So you basically start this process at the base of the pyramid, and then the collecting ducts all run toward the papilla, because by the time you get the urine into the collecting duct, you're pretty much done with it. So the collecting ducts are all going to dump into the renal papilla. Then it's going to be gathered in the minor calyx, the major calyx, and then the renal pelvis. So all of this process that we were just talking about, to put it into context, it is going to, let's go back to this figure. So your nephrons are going to either be all the way up in the cortex or kind of at the junction right between the cortex and the medulla. That's where all of the processes are going to go on. And then the collecting ducts are going to run through to the tip from the base to the papilla. And then you're going to gather the urine from the collecting ducts into the minor calyx, major calyx, renal pelvis, ureter, and then urinary bladder and urethra. So this is where you can follow the structure, and you'll be able to tell in this figure really easily the proximal convoluted tubule, descending, ascending, collecting duct. So this will help you to understand it, to label that. Okay, just a little bit of fizz to add to this. There is a structure that is associated right here, right next to the glomerulus, that has some important physiological function. It's called the Juxtaglomerular apparatus. Juxta meaning next to. Next to what? Next to the glomerulus. So the juxtaglomerular apparatus has this region that's sometimes visible when you draw a nephron and sometimes not. So afferent arteriole and efferent arteriole. But notice that this guy right here is actually folding between the afferent and the efferent arteriole, that's actually the distal convoluted tubule. And you can see it in one of these nephrons. Do you see in this one how afferent arteriole and efferent arteriole and the distal convoluted tubule kind of folds next to those two? Okay, so what happens when the efferent arteriole, efferent arteriole, and the distal convoluted tubule, when the distal convoluted tubule comes against the afferent arteriole, it creates this region right here, and this region is called the juxtaglomerular apparatus. And that's a really important physiological region. So basically what it has is there are cells in the wall of the afferent arteriole that are monitoring pressure, because pressure is really important because you're about to do filtration. And then there are cells in the wall of the distal convoluted tubule that are monitoring the chemical content of the urine because you're almost done with it. So what happens is the juxtaglomerular cells, which are in the wall of the afferent arteriole, they are pressure receptors, pressure monitors. If blood pressure is low, what they're going to do is they're going to start a mechanism of increasing your blood pressure. And what they do is they secrete an enzyme called renin, which will ultimately increase the pressure in the system. So if these detect low pressure, they'll say, we need to do something to raise the pressure. And that is secreting this enzyme called renin, which raises blood pressure by two mechanisms. And then there are cells in the wall of the distal convoluted tubule, and these are not pressure receptors, they're chemoreceptors. And what they do is they monitor the chemical content of the urine because you're almost done with it when you're in the distal convoluted tubule. Remember, the distal convoluted tubule is almost about to dump into the collecting duct, and then you can't do anything to it anymore. So if, for instance, there was like, hey, there's too much sugar in here, we need to really reabsorb the sugar because sugar is money, then the distal convoluted tubule would release some things that said, hey, there's too much or too little in this almost urine. So you've got one last shot and do something about it. So collectively, these two monitoring areas are called the juxtaglomerular apparatus. Now, you should be able to label them here. Now, the only thing I don't like about this figure is that the afferent arteriole and the efferent arteriole are not different widths or diameters, but remember that the distal convoluted tubule actually snuggles up to the afferent. So that's got to be the afferent arteriole, and that's got to be the efferent arteriole. So you can do all of that labeling right there.